I'd like to welcome everybody who's come to listen and take part in this special National Thrombosis Week session on COVID and clots. And this is really cutting edge COVID that we're going to cover today. We're going to cover some new approaches and also new ways of thinking and re-energizing how we used to think about thromboinflammation. To remind you that on Friday at a similar time, we also have another session talking about thromboprophylaxis in COVID patients and also covering VITS, which is vaccine-induced immune thrombocytopenia and thrombosis. So we will cover all aspects of COVID and thrombosis, but today it's cutting edge. And I'm very lucky to have three wonderful speakers. They've all got about 20 minutes and we'll have a discussion right at the end after a few questions, if there are any pertinent ones after each talk. So the first, we have Dr. Yogen Kanthi, and he's a rising star in US medicine. He's a Alaska investigator at the National Institute of Health, which means he's been picked out as a bright young thing to come along and look at research and take things forward. He's practicing vascular medicine, and he is a cardiologist at the National Heart, Lung and Blood Institute, and he co-directs the Vascular Medicine Fellowship Program at the University of Michigan Medical School. So he does translational research, seeking to understand how innate immune circuits communicate with coagulation, leading to inflammation and thrombosis. And he has done a lot of work during the COVID pandemic in this area. His long-term goal as a physician scientist is to develop new treatments for highly morbid venous disease, which, as we all know on this call, affects millions of people every year. So he's going to talk to us about current thoughts on the pathogenesis of COVID-19. Yogen, over to you. Thank you very much.
Wonderful. Thank you very much, Dr. Hunt, and to Joe and the National Thrombosis Week for inviting me. I've been following National Thrombosis Week now from the US for about 3 years, and I've always been watching it and jealous that I'm not able to participate actively. So I was delighted when Dr. Hunt invited me to be here. So I can assure you it was an easy flight in this morning from the US to London. I think you can see my slides here. Great, so I will go ahead and advance these. Wonderful. So first to just really acknowledge the team that's been involved in this. In a lot of our work, I think none of this would be possible without bringing together a large group of people, especially Ray Zhu, who is she. And I'll present some of our original research here today. And Jason Knights, who's my rheumatology partner at the University of Michigan. We also have a large clinical trial team that we assembled and a number of other collaborators. Finally, our funding sources, the National Heart, Lung, Blood Institute at the NIH, the Lasker Foundation, the University of Michigan, who really supported this work and funded it when we just had ideas and no data. So that was really kind and generous of them. So this group, I think, knows pretty well that inflammation and coagulation are very closely interrelated. In fact, we think that inflammation and coagulation are probably evolutionarily linked processes, so that when you have a wound, you can start to first stop the bleeding and then start to bring in the immune response in order to be able to fight off any microbes. And then you also need inflammation to help you heal. And it's no coincidence that these are certainly closely intertwined in both health and in disease. We know a lot of different cell types are involved in it. I'm going to just show you a brief cartoon of what we think happens in the blood vessels when people start to develop clots. Normally, this is the blood vessel itself. The endothelium is the lining of the blood vessel. It's a single layer of cells that are on the inside, and they sit between the blood and the vessel wall itself. We think it's more than just a blood wetting surface that allows fluid and blood to slip through. We think it actually is an active organ, probably larger than any other in the body. When the endothelium becomes activated, it starts to express a number of different molecules on its surface. And these molecules can do a lot of different things. They can trigger blood clotting, they can trigger immune responses. They certainly do start to recruit leukocytes or white blood cells from the bloodstream to try and stick to the endothelium and start to do some other signaling. Monocytes are a type of circulating white blood cell. They start to become activated, release things like tissue factor, clotting factors, cytokines like TNF-alpha. We found IL-1 beta is involved in clotting as well, and a number of other processes. Platelets, which we think of as kind of the bricks of brick and mortar in blood clotting itself. Platelets start to become activated, they aggregate together, start to release things like complement, which can then start to form a clot. And finally, this area called neutrophils. These neutrophils are the most abundant circulating white blood cell that we have. They're usually your first responders if you have an infection or inflammation or a wound of some kind. And they have this interesting thing that they do when they encounter bacteria or pathogens. They extrude or spit out this web of DNA, and this DNA actually forms a trap for bacteria and for viruses, and also serves as a scaffold on which blood clotting can happen. And so this is great if you're trying to prevent infection or contain things like sepsis, but these can be quite detrimental and deleterious if it's unrestrained. So we know that neutrophil extracellular traps, or nets, are certainly involved in thrombosis, especially venous thrombosis. And so I'll focus on that for the first part of the talk. I think it's always helpful to have a visual. This is an image that we did not generate. I'm sorry the credit doesn't show up. This is the cover of ATVB 2 years ago. The yellow is a neutrophil itself. This is a scanning electron micrograph in which they show the green extracellular trap that's been really cast out as a web from the neutrophil and is trapping the blue bacteria. So this is sort of a visual of what we think might actually happen within the blood vessel itself. So as the pandemic started in February and March of last year, we started to look at some of these reports, the preprints that were coming from China. And we noticed something that was quite interesting when we looked at this particular report, which was eventually published in The Lancet, and we compared survivors and non-survivors. These 54 survivors had profound lymphopenia, low lymphocyte counts, which I think has been really well described and consistent across a number of different studies now, and something we see in viral infections. But what was a little bit curious for us is that the white blood cell count was not low. In fact, it was higher than what we saw in the survivors or in some other normal healthy people. And so we wondered if perhaps neutrophils, these first responders, might actually make up the difference here, and whether these neutrophils could be doing something a bit insidious within the severity of this disease itself. So at this time, since we knew inflammation and clotting were so related, we thought to ourselves, it's surprising there aren't more clots. And certainly then we saw this report from Professor Beverley Hunt that she's never seen such sticky blood in COVID. There's a corona blood clotting mystery that starts to intensify. Then we saw reports from autopsy series of pulmonary artery clots, and this one from my colleague, Dr. Sharon Fox in New Orleans, of a number of different patients in an autopsy series who had clots in both the pulmonary and the cardiac vasculature. This is an example of the burden of thrombosis from Dr. Fox's paper. You can see wherever there's an arrow, there are clots. These are all the large vessels within the lungs itself of a patient unfortunately succumbed to the disease. When she zooms in on even the smaller blood vessels, I'll show you in a bit, their rampant clotting extends into those vessels as well. So the incidence of thrombosis is not low in patients with COVID. So if you're out of the hospital, we don't really know, and you're not hospitalized, we don't really know what your risk of clotting is. But if you're in the hospital, if you're sick enough to be admitted, and in some studies where they've tried to characterize it as best as possible by scanning people regularly, this study from the Netherlands suggested that up to 60% of patients in the ICU have a thrombus somewhere, either in the arteries or the veins. And another series that's at the bottom here suggests that if you're not in the ICU, it may be even as high as 11 or 12%. So certainly not a small number, and much higher than what we would expect for people who are admitted to the hospital on any other time pre-pandemic. So we started asking ourselves whether these neutrophil extracellular traps or nets could be involved in COVID. And so Ray Zhu went around the hospital, collected a lot of discarded serum from patients who were admitted specifically with COVID. At this point, our hospital in Michigan was essentially a COVID center, and we were in the process of starting to stand up field hospitals at some of the local stadiums. So when he measured circulating levels of NETs, what he found is that if you were in the hospital with COVID, you had levels of NETs and on room air, so not severe respiratory disease, you did have elevated levels of NETs compared with what we normally see in healthy patients. But on the other hand, if you were in the hospital with COVID and on a mechanical ventilator with respiratory failure, your levels of NETs were significantly higher. And those are the bars on the right of each of those panels. He also then took some of this serum from patients with COVID and wondered whether this could in turn start to feed forward into healthy neutrophils and cause problems there. And so he did some studies in which he incubated healthy neutrophils with serum from either healthy patients or COVID patients and found that the COVID serum had this ability to really trigger net formation even in normal healthy neutrophils. And here at the bottom right, you'll see a picture of this. The blue are the nuclei in the neutrophils, and they've got this multilobular nuclear shape. And the green itself is citrullonated histone H3, which is a marker of net formation. And so you can see it starts to really extravagate and form these really extensive networks that go across different cells outside its individual cell boundaries. And so certainly there are increased levels of NETs here. At this point, we looked back at Dr. Fox's paper and found that she had found that there are clots in the arteries and in the veins for patients with COVID. And also these clots were happening in both the small vessels and the big vessels. And so this is quite unusual. We hadn't expected to see this. It's not normal for us to find it across all four of those spectrums. But we have seen this in one other disease that I'm interested in, which is antiphospholipid syndrome, in which patients have these antibodies that start to bind to phospholipids or phospholipid binding proteins and can trigger thrombosis in the arteries and the veins and the small vessels and the big vessels. And so in some ways, it really did remind us of antiphospholipid syndrome. So at this point, we also looked back at some of our samples in which we had stimulated NETs, healthy neutrophils to form NETs with COVID serum. And found here, you can see the DNA, the nuclear stainings on the left in blue. On the top bar is COVID-19 serum. On the bottom is healthy serum. The green is neutrophil elastase staining, which is also a marker of NETs. And in the red here, you'll see human IgG. So we probed these NETs to see whether there were antibodies associated with it, and found certainly that in NETs formed by COVID serum, there are a lot of antibodies that are stuck onto the NETs itself. And so it starts to raise some other questions about what these antibodies might be doing. Or why they might be there. So I told you a bit about antiphospholipid antibodies. In some prior work, we've taken these purified IgG fractions from patients with antiphospholipid syndrome, incubated them with neutrophils. They bind to the neutrophils. They cause the neutrophils to undergo this process of an uncontrolled explosion where the DNA webs are cast out. And we certainly know that these can trigger thrombosis in antiphospholipid syndrome as well. So we decided to look at antiphospholipid syndrome or antiphospholipid antibodies in our patients with COVID. But before that, we started to see some reports come out of APL antibodies being present in patients with COVID. Now, at first, we thought perhaps these were, quite limited size case series, and so there may or may not be anything to be made from this. And we decided to look at our own cohort of 172 patients. What we found was interesting when we measured 3 different types of antiphospholipid antibodies: cardiolipins, beta-2 glycoprotein 1, and then antibodies targeting the phosphatidylserine prothrombin complex, which is a bit of an outlier. It's not something that's usually done in clinical practice, but we use it sometimes in patients who are otherwise seronegative, and found that if you just look at the normal cutoffs, about 50% of the patients had an abnormal antiphospholipid antibody of some type or the other. And in fact, when we use a more stringent cutoff, which is what rheumatologists and hematologists typically use to diagnose somebody with antiphospholipid syndrome, it was about 1/3 of the patients. So still, again, a lot higher than what we would expect to see. But these antiphospholipid antibodies have been noted before in patients who have other illnesses like sepsis or acute respiratory distress syndrome. And so we wondered whether these antibodies were actually functionally active in doing something, or whether they were just a correlative biomarker for the severity of illness. So we looked at the clinical parameters for patients who had abnormal antiphospholipid antibodies, and they seem to correlate with this SpO2-FiO2 ratio, which is a marker of oxygenation. So the lower the oxygenation, the more the presence of antiphospholipid antibodies. We also noticed that there was an increase in the amount of circulating NETs, higher levels of D-dimers, and also an abnormal level of calprotectin in patients who had abnormal antiphospholipid antibodies. And we found that this calprotectin may be a better biomarker to measure in people who are admitted to the hospital on the first day or two, and be able to predict better than anything else that we found so far, whether or not patients are going to end up with respiratory failure on the mechanical ventilator. And I'm glad to be able to say this, that there's a company that's trying to move this forward into the clinical arena as a diagnostic study. There's also a group from the UK, from London, that's actually received funding to be able to develop this as well as a biomarker to move into the patient arena. So we're really glad to see that move forward. But we did ask ourselves whether these antibodies were doing anything to the neutrophils themselves. So we took IgG fractions from the circulation and we incubated of patients with COVID. We incubated them with healthy neutrophils and found that they are able to trigger net formation again. This is what it looks like in the white bar if you don't have COVID IgGs. But when you add COVID IgGs, you see a 2.5-fold increase in the amount of netosis. Then we added a drug called dipyridamole, which is an antiplatelet adenosine-stimulating drug that we've tested in net formation before. And when we add that, we can reduce the amount of net formation by about half. So there's a modest but important improvement in the amount of netosis that these antibodies can trigger. To see if this actually might lead to a clot, we injected animals in a mouse model of deep vein thrombosis with IgG fractions from patients with COVID. And what we found was fascinating here. In the blue circles in the first bar, that's what a normal amount of clotting looks like when healthy IgG is given to mice. And so that's about 4 to 6 millimeters in size. But when we injected mice with the COVID IgGs, we found a massive amount of thrombosis, 2.5 times the size of what the normal clots look like. In fact, this is the worst clotting we've seen in mice before. So it certainly is something we won't forget. And it does make us wonder whether these antibodies are, and actually suggests to us that these antibodies might actually be driving a clotting process itself. But there are other components in the bloodstream. I've told you about neutrophils and autoantibodies. Platelets also are quite important. They, again, are part of the bricks for the brick and mortar aspect of thrombosis. And in patients who have COVID and either are in the ICU or not, the number of platelets doesn't seem to change all that much. But these platelets are actually, interestingly, much more talkative. They communicate and interact much more with leukocytes like neutrophils, monocytes, and T cells to be able to perhaps drive an immune response or a hyperactive immune response. And then also these platelets are actually quite sticky. So what you'll see here on the right is a graph that shows how much aggregation platelets have, and that's what we need to form a clot. And in the red line, you can see that those are the COVID platelets, or patients from COVID from patients with platelets that get a maximum amount of platelet aggregation that you can measure. It's 100% really with any minor stimulus. But the healthy platelets will actually have a normal small amount of aggregation. And so these platelets in patients with COVID also seem to be quite hyperactive. So thinking back about the other aspect of the vasculature that might be playing a role here, the endothelium, again, this lining and interface that maintains blood fluidity between the blood and the blood vessel is something that we think is an active organ. And we know that in patients I apologize, this is the wrong slide. Let me move to the next one here. We know that if you take healthy endothelium and you expose it to COVID serum, it wipes away this protective anticoagulant layer of heparin sulfate that sits on the endothelium and helps maintain that antithrombotic effect. And certainly there are reports here that heparin sulfate may be playing a number of different roles. And I think the speakers that will be later in the session you'll hear from will share a little more about heparin sulfate. In this report from another one from Dr. Fox's autopsy series, she shows that the endothelium in both the heart and the liver is actually somewhat disrupted, and there's this cuffing around it in the yellow arrows I show that's made up of inflammatory cells, of leukocytes like neutrophils and lymphocytes that may be surrounding the blood vessel and perhaps informing some of that inflammatory signaling cascade that we're starting to see. And so Hui Shi in our lab decided to look at try and understand what else might be happening with the endothelium besides wiping away of that anticoagulant layer. So she incubated serum from healthy patients or people with COVID with healthy endothelial cells and measures these cell adhesion molecules on the surface itself. So these again are the molecules that start to really come out, and then almost like Velcro, they adhere to the leukocytes and start to facilitate more of that inflammatory and immune crosstalk. And what she found here was quite interesting. She found that there was an increase in the number of these cell adhesion molecules, but also she actually observed that the leukocytes, in this case, the green are neutrophils that might be stuck to the endothelium, neutrophils will actually stick very, very tightly, adhere to the endothelial layer following treatment or exposure to COVID serum. So certainly there's a multifactorial approach or multifactorial system here that's really informing inflammation in this context. What she found here also is that the cell adhesion molecules, in particular E-selectin, VCAM, and ICAM, were significantly elevated. So I'll pause here just to summarize that circulating, we think that thrombosis is mechanistically a multifactorial process in the context of COVID. Circulating IgG antibodies do start to activate the endothelium, they activate neutrophils. A group in Germany has shown that these IgG can also activate platelets and cause them to start to become hyperaggregatory. We know that circulating antibodies and also NETs itself are associated with respiratory failure in patients with COVID. And I'll skip this anti-net IgG part because I think I haven't shown that to you yet. In future work that we're doing now, we're trying to understand what might drive the production of these autoantibodies. Importantly, are these antibodies that are normally present but in very low levels in healthy people? And then perhaps in the setting of some sort of an acute illness, a severe illness, the brakes that help suppress this antibody response are maybe released and we start to see an exuberant production of them to be able to do a number of maladaptive things. And then also, do these antibodies go away after the infection or after the acute insult? Or do they persist in some time in the course of recovery? So we're examining this now to see whether this happens in patients after they've left the hospital, and especially those who are now coming back and donating convalescent plasma. So there's a lot yet to be done. We know a little, we're understanding more. This is informing us not only about this disease, but also principles that may be about human physiology that may be important and have applications to other diseases when we're on the other side of this pandemic. So I'll stop here and thank you for your time and attention, and cede the mic back to Dr. Hunt and the other speakers. I'll be glad to take questions at the end when the Q&A session starts. Thanks.
Thanks very much, Yogen. Do you want to take a few questions now?
I'd be glad to.
Okay, so I mean, I do think that we are seeing the stickiest blood ever in these patients, and we've just published some work where we looked back at patients who required ECMO with COVID and compared them with other patients who required ECMO for viral pneumonias over the last 8 years. And when you look at them, we do a CTPA before we start, and they all had a higher rate of immunothrombosis and PE on their scans when they started. And then at the end of ECMO, we checked to see if they have any DVT, and they had a much higher rate of DVT with COVID than with any other viral pneumonia. And so your work is absolutely fascinating. What do you think about the antiphospholipid antibody here? Because we do see transient antiphospholipid antibodies with lots of infections. And the early reports in the New England Journal saying, well, everyone's got lupus anticoagulant is because if you've got a raised CRP, it'll interfere with the lupus anticoagulant test. So your data suggests that they are actually causing harm. Do you think that they're causing harm, or are they an epi harm?
This is a great question, and I think I really commend you and congratulate you and your team, because the UK has really been at the forefront of being able to actually have a systems and infrastructures in place to be able to try and study these processes in a very organized way. And so the contributions, I think, in not just in the diagnosis of the disease, but understanding some of the mechanisms and the correlative science, as well as the clinical trial infrastructure, has really been, I think, a tremendous service to the global community. So congratulations certainly on that. We think that these antiphospholipid antibodies may be really at least in part a canary in the coal mine. They may be an indicator of an underlying unrestricted B cell response in which antibodies are produced to a whole host of self-directed antigens, and we don't know what those antigens are doing. Perhaps the amount of antiphospholipid antibody we detect is a biomarker in some ways of what the other antibodies might be doing. But we do know that antiphospholipid antibodies on their own can trigger thrombosis. And so perhaps this is both part of the part of the larger repertoire and a biomarker for the repertoire, but perhaps they are also serving as a procoagulant in some ways. And so I think it's going to be difficult to separate the two. We are trying to do that now. But I think it will be important to try and understand what else might be happening. So in your cohort of patients, measuring the amount of antiphospholipid antibodies would be informative. It would tell us. So was that something you've done already or hoping to do in the future?
So you have described how much research we have been able to do, but our research has been totally regulated in the UK by our chief medical officer. So he actually only allowed certain areas of research. So if you wanted to do a project, you had to get it okayed by him, and he had a huge backlog, and he only okayed the really big clinical trials. So we haven't been able to do things like that, which you have had a free hand to do. So congratulations to you for taking the opportunity there. I'd like to come back to our discussion at the end, but I think I have to move on now to the next speaker. And this business about, in the UK, we did some good research, but we had to focus down on what we're doing, is very pertinent to the next two talks. So first we've got Andy Webb, who I work with at Guy's and St Thomas's, talking about heparin as an anti-inflammatory and antiviral agent. I've known Andy for a very long time. He's a senior lecturer and honorary consultant physician in cardiovascular research in our clinical pharmacology department here, and also London British Heart Foundation Centre of Research Excellence. His research is related to blood pressure and hypertension and cardiovascular function. And he was exploring nitrate and nitric oxide pathways in experimental medicine studies before he started off with COVID. And he has led on an application for trials of inhaled heparin for early treatment and prevention of COVID. And he's going to talk to us about heparin as an anti-inflammatory and antiviral agent. Over to you, Andy.
Thank you very much, Beverley, for that lovely introduction. And thank you to Joe as well for inviting me to talk. I'll try and share my screen. There we are. Does that show? Okay, great. Okay, thanks. I'm going to put my timer on as well, so I have an idea of the time. So thank you very much. So this, in normal times, is almost my view from the clinical research facility where my office is. But over a year ago, of course, all our clinical non-COVID studies shut down. And hopefully they'll restart again in the near future. And the unit was taken over by the Oxford AstraZeneca vaccine trials, which of course have been incredibly important. So I wanted to sort of just have a reflective approach at first. So I remember when this all started, as we all did, and this was this terrifying photo of Dr. Li Wenyang, who then died on the 6th of February. And he was rather young, he was 34. And so I remember being pretty worried about this disease. But I thought, well, as with other pandemics and things, we'll probably leave it to the infectious disease doctors, virologists, public health, and just carry on. So I sort of perhaps sleptwalked for a bit, and perhaps I should have stuck by my initial instincts, but then I had an email. I can move this on. There we are. On Friday the 13th from a colleague about the use of angiotensin receptor blockers, and does this explain that patients who take these with cardiovascular disease, hypertension, etc., do worse? Is this through an effect on ACE2 and also the binding of SARS-CoV-2 to ACE2? And so I then, a couple of days later, the Ides of March, developed a cough, breathlessness, purple toes, so I had to go into self-isolation. And so I had time to sort of ponder and look into this. And of course, this was the famous model that came out, or diagram that came out around that time in the New England Journal of Medicine. The traditional view on the right of ACE, also ACE2. In the sort of hypertension world, we've been a little bit interested in this in the past, with the different effects of ACE inhibitors, angiotensin receptor blockers, direct renin inhibitors further up the pathway as well, and the different effects of blocking the AT1 receptor, for example, versus blocking higher up in different positions, and what other products would be produced, and including angiotensin 1 to 7 on the MAS receptor. So all of that sort of stuff, but that's a bit old. But of course here we've now got the virus binding to the receptor and then being internalized. But most of this didn't give it away, and it wasn't until I read a little bit more, and this was the sort of the simple word association game, that the coronaviruses bind to ACE2, and this is a type 1 integral proteoglycan. And before it binds to that, or as it's binding to it, it also docks on these docking proteoglycans. So it was literally a small step to think, well, heparin displaces things bound to proteoglycans, and can prevent, therefore, things binding. And so, therefore, could heparin displace the virus and be a prevention or a treatment? And this was the sort of model that I found subsequently, and this goes back to the previous SARS, binding to these docking heparin sulfate proteoglycans to then find ACE2 and then be internalized and infect the cells. I was aware of this effect through work I'd done previously, part of my PhD on xanthine oxidase, and this is only the second time I'm presenting at a haematology conference. This was the previous time back in Manchester in 2009 at the British Blood Transfusion Service and the Red Cell Special Interest Group. But xanthine oxidase, of course, has a major role in gout. And also at that time, I was aware of this really nice paper by David Harrison's group. This is Speakerman et al., where they gave healthy volunteers 5,000 units of heparin, had quite a substantial increase in circulating plasma xanthine oxidase. So the heparin displaces the xanthine oxidase into the circulation. And this is patients with coronary artery disease, and here a massive increase, probably about a tenfold or so increase in circulating xanthine oxidase. I was interested in xanthine oxidase, so this is a bit of a sort of aside, but this shows how I sort of got onto its antiviral effects, because xanthine oxidase could be a nitrite reductase. So I was interested in nitric oxide, also previously known as endothelium-derived relaxing factor, and this was an alternative way of making nitric oxide in the circulation. And so that's the sort of model. And I looked in heart tissue, also blood vessels, subsequently, and this is left internal mammary artery, and this is adding allopurinol, looking at nitric oxide production from nitrites, which is partially inhibitable by allopurinol, the enzyme inhibitor for xanthine oxidase at this molybdenum site. We also did some immunohistochemistry staining and found xanthine oxidase on the surface of the red cells as well. So hence partly this presentation at the Red Cell Special Interest Group back then. But whilst I was collecting that tissue, from patients undergoing coronary artery bypass grafting, we noticed, and this is a common finding at that time, that blood pressure drops about 5 minutes or so after the big heparin bolus, and patients get about 20 to 30,000 units of heparin just before they go on to the perfusion. And so I also set up a xanthine oxidase assay and collected the blood, and there's a massive increase at around 10 minutes, so similar to the Speakerman paper I've shown you already. When I was aware again that SARS-CoV-2 bound to ACE2 and it's a proteoglycan and it docks on the proteoglycan, I thought, well, surely this is easy. We can knock it off with some heparin. So that was the question. I looked back and found this paper I partly presented already on the first SARS virus, and there was some evidence that heparin could indeed do that. And so this was the dose response. You go up on the concentration of heparin, you can reduce the infection of human and mammalian cells. And so that's the mechanism I've sort of explained already, but elimination of cell surface heparin sulfate by exogenous heparin, reducing the ability of the virus to bind to the cell surface, increasing cellular resistance to infection. And so this is the sort of model. I've slightly adapted the one here, but by putting heparin on here, binding here, you'll hopefully stop the virus binding. So a really simple but specific mechanism, potentially, if it works, of course, for SARS-CoV-2. And then this paper came along on the end of March, and and published in Nature, showing that actually the way that SARS-CoV-2 here in blue at the bottom binds to the receptor ACE2 is itself via glycan, shown here in red as well. So this may also be amenable to blockage via heparin. And then in parallel, so this was from the 29th of February, this is the group with Mark Skidmore and colleagues. So they were interested in actually heparin binding to the spike protein and seeing whether that actually results in an alteration of its capacity to bind to the receptor. And they found evidence of that. So here was an additional mechanism, an amazing additional mechanism as well. So it all looked quite exciting. But then we had to decide on if we were going to consider using this in a study, what the route of administration would be. And because obviously we're dealing with a respiratory virus, could we give it for the lungs? Is ACE2 expressed in the lungs? Yes, it is, and several old papers showing that it is. So could we give it to the lungs? Can it be inhaled? And initially I had no idea. I've never heard of inhaled heparin before. But a quick PubMed search showed yes, it can be. And look, there were, this was a systematic review I took a year ago, 39 human studies at that time, over 1,200 patients, 8 randomized controlled trials. And many of these were performed in asthma, 15 trials, also burns and inhalation injury. So it's been quite fairly widely, not massively, but fairly widely trialled in these. Ventilator-associated lung injury, cystic fibrosis, COPD, pulmonary fibrosis, and a few healthy volunteer studies, including pharmacokinetic studies. This was the median dose that had been used, 60,000 units, that's the daily dose, and even up to 400,000 units of heparin was clinically feasible, it was reported, without significant clinical events. No significant statistically significant bleeding risks were found either. And a few isolated cases of hemoptysis, but they appeared to resolve quite quickly with treatment withdrawal. And not much evidence of harm. I'll skip over this in the interest of time. So I wrote to Professor Beverley Hunt on the 18th of March and sort of thought, should we try and do something? Systemic heparin trials, I think, were already being proposed at that time. So we thought we'd see if we could put in a bid, a grant application for inhaled heparin. And at that time, there seemed to be very little else going on around the world that we're aware of, at least. And actually, this is a substance search I did in June, and there were only these 2 trials, both on ITU, I think, patients. This was the larger charter trial from Australia for ventilated patients. So moving on, so really this was a specific mechanism, is effectively an antiviral mechanism. Where I saw it at the time, any anti-inflammatory mechanisms would be secondary to the antiviral effect. So this is a diagram I've sort of adapted for heparin. So you have for heparin, you can block the binding to the cells, the subsequent inflammation, but also the replication and this cycle. You could interrupt this vicious cycle by giving heparin and perhaps therefore reduce the spreading inflammation across the lung surface and the secondary inflammation that ensues. This sort of model was available almost exactly a year ago with the inflammation spreading through the lungs as compared to bronchopneumonia, for example. Here's ACE2 on the type 2 pneumocytes. And as we've heard very nicely from the previous speaker, the immunothrombosis in the lungs, heparin surely might also be a bit useful for that, or at least not deleterious, we'd hope for that as well. This model as well shows that the downregulation of ACE2, initial ACE shedding, called ACE1, and with the resulting increase in angiotensin 2, which itself can have multiple actions, NADPH oxidase, for example, oxidative stress, inflammatory cytokine release, and therefore creating this pro-inflammatory environment partly through this mechanism, but then potentially later in disease, with a loss of ACE, you're then getting a reduction in angiotensin 2, which might be necessary to prevent excess vasodilatation, vasodilatory shock. And another aspect of ACE2 is that it's also involved in bradykinin and the inactivation of desarginine bradykinin. So here's the ACE2 down here at the bottom, So inactivating this, so with a loss of ACE2, there may therefore be an excess upregulation of this inducible B1 bradykinin 1 receptor, also resulting in inflammation, besides the other mechanisms that we've already discussed. And to put this in context with the coagulation pathway, of course, the bradykinin system sits alongside this and also complement. And also here we've got the downstream signaling from the B1 receptor, MAP kinase, etc., NF-kappa B, going through to INOS and nitric oxide, and then the B2 and the pathways there as well, just to put that all in context. I think our next speaker, Quentin, might discuss this, and he's a co-author on this very nice paper. This is by Frank Harin and colleagues, published in the summer or so last year. So we've already covered some of this, but then moving on to the other inflammatory pathways. So there's activation of macrophages. We've already heard about a bit in the previous talk. This inflammatory exudate rich in plasma-borne coagulation factors entering the alveolar space, followed by expression of tissue factor by alveolar epithelial cells and macrophages, the formation of fibrin in this hyaline membrane as well. Nets, we've already heard about, so very nicely described. These neutrophil extracellular traps composed of the DNA cytotoxic histones and neutrophil elastase, causing further lung injury. And also the microvascular endothelial damage that we've already heard about. So the idea here in this diagram is that if you to inhale heparin, this unfractionated heparin, UFH here, it would flush it all away miraculously. So there we go. And these are some of the mechanisms described here. It's going to, as we say, prevent the binding to ACE2, reducing this hyaline membrane formation down here, microvascular thrombosis, counteracting hyperinflammation, the formation of nets, increasing NO release, possibly with vasodilatation, and also having some mucolytic properties. So some further mechanisms are included here. So Jan Shute and also Barbara Malloy previously showed that heparin forms complexes with IL-8 and also IL-6, rendering them unable to activate the receptor or bind to their receptor in the case of IL-602. Also resulting in reduced chemoattraction of neutrophils into the lung following heparin treatment. Inhibition of neutrophil-related activity through neutrophil elastase and its release, neutrophil aggregation, cathepsin G. So a whole range of potential downstream effects. And we've also already talked about DNA neutral nets. Inhibition of complement activation, shown by Barbara Malloy as well, and inhibition of pulmonary fibrin deposition. So multiple downstream mechanisms. So we put them together. I'm just going to check the time, 15 minutes. This application, this went in, this was Easter Sunday last year, and Beverley Hunt and other colleagues also show this by way of acknowledgement to the team as well that we put together to put in 2 trial applications. One was prevention for COVID. This was in frontline health care workers, and other people who might be exposed as frontline staff, and also treatment for early treatment on admission to hospital with inhaled nebulized heparin. I've not included the protocols here in the interest of time, but that can be discussed, and I know Quentin's going to talk more about some trial designs, I think. So we got through that first stage, invited to apply for NIHR funding and a full application. The hypothesis I've sort of already described, so in the interest of time, I think I'll skip over that, the mechanisms we've discussed. And so we were preparing to put in the next application, about to put it in, and one of my colleagues sent this email, 1st of June, 9:10 a.m. I read about nebulizer heparin in The Guardian this morning, and this was the article, here we go, Monday the 1st of June, UK hospitals try 5 new drugs in search for coronavirus treatment. And this was the Accord trial, which we were very familiar with, but heparin hadn't been added at that stage. And then that was then added, and this was going to be trialled at St Thomas's, where Boris Johnson, our Prime Minister, of course, was treated, and Guy's in London. The 5 drugs which can be disclosed for the first time are heparin. That was there. So that led me to contact a colleague, Professor Clive Page, in pharmacology, which is slightly separate to clinical pharmacology where I'm based. And he'd been working on this for a long time. And I hadn't been aware, embarrassed to say. And so then we liaised, and also I found myself on a call with Professor Tom Wilkinson, respiratory consultant in Southampton. So we sort of paired up. I dropped our treatment trial application, so there was no competition with the Accords treatment trial. And with Clive Page now on board and his expertise, we submitted our prevention trial application the 11th of June. Unfortunately, a month or so later, the application was declined. And of course, by that time, COVID was sort of come and almost gone, and perhaps that was the end of the need for inhaled heparin for COVID. Of course, there was no vaccine at that time, but then there was the second wave in November, and Beverley Hunt and Dr. Andrew Webb, an ITU consultant at Guy's and St Thomas's, presented with the patients being admitted to ITU and going on to ECMO were younger and sicker than in the first wave. And I'd linked in with Clive Page and Frank Van Haren in Australia. There were, as I said, no vaccines at this time. So we thought, oh, should we do something? Sort of here we go again. Do we really try again? But with the second wave, it also brought good preclinical data. And this was particularly on the antiviral effects that I was interested in, sort of started with. This is the team, Julia Tree from Porton Down, who've just received a large grant today, and showing more convincingly this dose-dependent inhibition of, this is viral plaque, and also this binding. So heparin also binds and stabilizes the receptor binding domain of the spike protein, directly inhibits binding to human ACE2. And so here we've actually got quite good IC inhibitory concentration, 50% for both of these effects, which are quite similar across several different types of heparin here. And it's estimated, this was the estimated concentration that you'd get in the airways if you nebulized heparin. And so the IC50 is considerably below the concentration that's achievable in the lung. So this was, so when I saw this, I thought, right, okay, 30th of October, it's worth pursuing this. This sort of data was replicated in very nice studies. You could achieve, with this concentration, 80% inhibition of invasion of mammalian virial cells, and similar data on inhibition of those virus docking to the proteoglycans. Where is the ACE2 mainly expressed? It's mainly in the epithelium, and so this epithelium is shown in blue, particularly in airways, also kidneys, but many of these are airways. And the endothelium is present in some of the vascular endothelium, but not detectable in others. So I think by giving it to the lungs and respiratory epithelium is probably the right approach. And this is a nice study performed partly in Trieste and also with Mara Jacka, who joined our cardiovascular school a few years ago from Italy. And this is a post-mortem series. But the sort of take-home message here is that even 30 to 40 days from diagnosis, there's evidence of the virus in the lung pneumocytes, and also the spike protein. So it really hangs around. It also causes this derangement of the cells. Here you've got a cell with 2 nuclei, rather large, it's got an inclusion body, but even up to 20 nuclei. So really bizarre pneumocytes are being produced. So it's having an effect that's not only through inflammation, but also by creating these dysfunctional cells. I'm probably running out of time. So yes, I'm just over 20 minutes. So the last couple of slides, human pharmacokinetics, it hangs around in the lung if you inhale heparin. So half-life estimated about 28 hours. So that's useful if you want to have an ongoing effect in the lungs. So the summary here is, I won't sing this, but well, this is automatic. Against almost any coronavirus that binds to heparin sulfate proteoglycans and ACE2. It would include variants and all the new variants that we're concerned about, an alternative to vaccine if there's resistance to the vaccine. Of course, we need the evidence to see that it works. It's systematic. It's got this series of effects: antiviral, anti-inflammatory, anticoagulant, mucolytic, perhaps antifibrotic even for long term. It's hydromatic, if I can use that expression. You can put it in a solution and nebulize it. It's nebulizable. So why it's inhaled heparin. So it's in international meta trials, and I think Quentin will talk about that. We've got in a couple of UK CTAP applications currently. This is with apologies to Greece, of course, and Greece Lightning, and perhaps some juniors who might be too young to remember where that comes from. But go heparin. Thanks very much.
Oh, thanks, Andy. That has been very magical, because you took us all the way through from the beginning of COVID and trying to get the research going. And it's really an answer to Yogen that we tried so hard to get research going and we were blocked by the top. They only wanted the really big platform trials to be operating within the UK. I think I'm going to move on to Quentin Nunes, and we'll have questions for both of you after you finish, because I think you naturally flow into each other. So I'd like to introduce Dr. Quentin Nunes. It is just so lovely to have a consultant hepatobiliary and pancreatic surgeon here in National Thrombosis Week talking about heparin. It's a first, and it's a very wonderful first. He works at the East Lancashire Hospitals. He focuses on looking at heparin binding proteins in disease. He's had National Institute of Health Research grants, and also the Royal College of Surgeons has supported him. He leads in hepatobiliary surgery, and he's an honorary senior lecturer at the University of Liverpool, and he's a clinical trialist, and he's going to tell you about the clinical trials he's tried to get going around inhaled heparin. So fantastic. Thank you so much for coming, Quentin. Over to you.
Thank you, Beverley, for that lovely introduction. And thank you, Joe, as well, for giving me this opportunity to speak at this session this afternoon. So I'll give you a brief overview of the work that we've undertaken, some trials that are being done at the moment. And I'll probably try to skirt some of the topics that Andy and Yogen have covered. So I'm going to speak to you today about inhaled heparin and some of the learning and research. So can you hear me? Hello?
Yeah, yes.
Can you hear me? Okay.
Yes.
Sure. Okay. So coming to my first slide, I'll tell you a bit about heparin sulfate and heparin. So heparin sulfate is a glycosaminoglycan that consists of repeating disaccharide units of idiuronic acid or glucuronic acid along with N-acetyl glucosamine. And it's a very important molecule that is present at the cell matrix interface and plays an important role in cell communication by virtue of binding a number of proteins at this interface. Now, heparin is an experimental proxy for heparin sulfate, and it's widely used as an anticoagulant, as you're aware. Now, heparin is a highly sulfated derivative of heparin sulfate, and in the clinically available form consists of a number of polysaccharides of varying lengths and molecular masses ranging from 5,000 to 50,000 Daltons. In the aqueous form, it is very stable, and that's very useful for us in clinical practice. It's mostly derived from porcine sources, but in some countries, heparin is derived from bovine sources and is in use. So heparin is probably the most negatively charged molecule known in biology. And by virtue of its charge, both heparin and heparin sulfate bind to a number of bioactive molecules. These are the heparin binding proteins, and they form very highly interconnected functional networks, which are important in health and disease. And this was some of the research that I did during my PhD and postdoctoral work over the years. Now, heparin binding proteins bind to heparin and heparin sulfate mostly due to the charge associated with both. However, there are researchers who would argue that there are specific heparin binding sites on these proteins by which they bind to heparin or heparin sulfate. And the group I work with has done some pioneering work in this field and developed some novel assays which are linked to arginine and lysine residues at these binding sites. And this technique is called a protect and label technique to identify heparin binding sites in these proteins. Now, more work is needed with respect to heparin binding proteins to better elucidate how heparin affects its non-anticoagulant roles in health and disease. And that's where it's particularly relevant in COVID-19 and other emerging viruses. So when it comes to heparin in COVID-19, I think as some of the speakers have already alluded to, in early 2020, we had a lot of anecdotal reports on the internet and in literature, which alluded to the possible potential beneficial effects of heparin in COVID-19 treatments. Around that time, the group I work with, along with some other groups, published data that showed that heparins, whether the unfractionated or the low molecular types, actually inhibited cellular invasion of the virus. And this was mainly by changing the conformation of the spike protein on the virus, which prevented cell invasion. Now, that was in early 2020 and attracted a lot of attention and collaborations. As some of the speakers have alluded to, heparin has potential anti-inflammatory effects by virtue of binding to a number of anti-inflammatory and pro-inflammatory cytokines and inhibiting them. It has local anticoagulant effects, inhibiting clot formation locally, and has some mucolytic effects as well. Now, all of these are useful in COVID-19 associated lung injury. Now, I know there are many studies that are investigating heparin administered by various systemic routes, but I'll focus on inhaled or nebulized heparin. So nebulized heparin has particular advantages in that it distributes heparin uniformly in the lungs. The pharmacokinetics are well described. There's very little systemic anticoagulation in the doses that are frequently used in nebulized heparin therapies. And nebulized heparin is very safe, with not many significant side effects described. It's important to know that, note that nebulized heparin is not licensed for use in the UK. So while heparin is a well-used and well-known drug, it is not licensed for use in the nebulized form in the UK and in many countries. However, it has been extensively investigated over the past decade or more. And it's a cheap drug, which is safe and easily available, and therefore it's particularly useful. Now, coming to the evidence of nebulized heparin in the preclinical setting, there have been a number of preclinical studies that have shown that nebulized heparin is particularly beneficial on its own, as well as in combination with other drugs in acute lung injury models, murine and otherwise. And I'll skirt through these various studies, but essentially they showed that nebulized heparin decreased pulmonary coagulation, decreased inflammation in the lungs, and basically improved oxygenation in these models. There's also a lot of clinical evidence for the use of nebulized heparin in ARDS and other lung diseases. And again, most of them show that nebulized heparin has got significant positive effects for patients with these diseases and in ARDS, improving oxygenation, decreasing the number of days on mechanical ventilation, in some cases preventing intubation in these patients. And a recent study by Barry Dixon, who's a leading intensivist in the field of nebulized heparin from Australia. And this was a pre-pandemic study looking at 256 patients who were on mechanical ventilation. who were either diagnosed with ARDS or at risk of developing ARDS. And he showed that nebulized heparin in the dose of 25,000 units every 6 hours for 10 days actually resulted in a significant decrease in the deterioration of the Murray score for lung injury, as well as it increased the proportion of patients at home at 60 days. So a considerable benefit from nebulized heparin in ARDS. And so there are a number of studies that have actually sought to investigate nebulized heparin in COVID across the globe. And I'll only list some of them and our local perspective on it. So we've got the NEBU-HEPA study, which investigates nebulized unfractionated heparin at a dose of 5,000 international units every 8 hours. And this is along with systemic enoxaparin in a prophylactic dose. The primary endpoint was intubation in this case, as was with the next study, which is NAIL-HEP, which looked at unfractionated heparin through the nebulized route. But the dose was slightly variable because this was a global multi-centered study. And depending on the local practice and the facilities available, this looked at pre-ITU patients in a hospitalized setting. Again, the primary outcome was intubation. Now, coming to the ACCORD study, which Andy alluded to, this was an important platform study in the UK. And initially, nebulized heparin, the unfractionated variety, was included in one of the arms that was being investigated. And this was a significant development for us in the UK. Unfortunately, this arm was discontinued for funding reasons. In terms of the other studies that are being undertaken worldwide, Frank Van Haren and Barry Dixon from Australia have been leading on the Charter Meta trial. Now, this study looks to investigate unfractionated heparin given in the dose of 25,000 units every 6 hours for 10 days. And this is a meta trial of a number of centers across the globe. There have been significant challenges, as everybody is aware of, with research into COVID-19. And I think this was a rather smart way of getting teams across the globe to work together. The primary outcome was a composite hierarchical outcome, which was alive and ventilator-free score at 28 days. In the UK, we were part of this collaboration, and we sought to undertake the same study at multiple centers in the UK, along with Professor Hunt and Andy Webb and Clive Page, and with collaborators from Liverpool and a number of other centers. Unfortunately, we were not successful in obtaining funding for this study, and one of our applications is still pending with UK CTAP. So, in summary, I'd like to say that heparin is versatile, it's safe, and relatively cheap, which has important implications for pandemics of this nature, because it is affordable to low and middle income countries as well. The inhaled route affords advantages, particularly in respiratory diseases like COVID. Now, further research is needed because while we have the nebulized route, which is probably the most widely used route at the moment to deliver nebulized heparin and inhaled heparin, it is less efficient. And there's more research that is underway to develop further devices to deliver heparin effectively. So we need more research in terms of devices and delivery. Another important aspect, and while this is a summary slide, I think I should delve on it here. We used unfractionated heparin because it's a very crude molecule, and because it binds to a lot of bioactive molecules. But there's more research needed to develop modified heparins that are more specific in their action. And the reason I say this is because there's not enough information in this field. And again, this is a summary slide, but I'm still going to delve on it, is because heparin heparin treatments are like double-edged swords. While heparin can bind and inhibit a lot of molecules that are responsible for disease and making conditions worse, there's not enough information out there because it can also bind protective molecules. So there's not enough information, and context is very important in the use of heparin. In this respect, it's very important that we have further research into heparin binding proteins, because I think research into heparin binding proteins will actually give us and elucidate the mechanisms by which heparin affects its non-anticoagulant activities. And so I think this is very important going forward, because there is research out there, some research from our group as well, to show that heparin binds a lot of viruses, inhibits a lot of viruses, and I think it is an interesting molecule to be investigated in the future. So I'll end there. Thank you again for your time and attention.
Well, thank you very much, Quentin. So we have another view about the use of nebulized heparin and more evidence that how regulated we are as far as the clinical trials that we can do in the UK, certainly during a pandemic. And it's a shame we haven't been able to get one going. And I can promise you it's not from lack of trying, especially Quentin, who must have spent many hours working on the grants. So I'd like to ask all the speakers to light up their videos, and let's have a discussion. Yogen, do you think you could get trials of nebulized heparin going in the US? Is the obvious question to ask.
Yeah, this is a it's a great question, and I really enjoyed both Dr. Webb and Dr. Nunes' presentations. I was in the chat box quickly messaging Andy and telling him how much I was learning about it. And I'm an angiologist, a non-invasive vascular medicine person, so it's and I understand endothelial biology to some extent. So and I was still learning a lot. So it's always helpful. In the U.S. we've also struggled a little bit to try and move some of these mega trials forward. As you know, the NIH, the National Heart, Lung, Blood Institute in particular, has supported this multi-platform trial to be able to test the role of anti-therapeutic anticoagulation in patients who are hospitalized. And certainly they were able to do that in collaboration with REMAP-CAP and the ATAC in the UK and the ATAC group in Canada to be able to get enough of a signal. And I think those studies are going to be coming out soon. That to me would sort of be the right place to consider interventions like this. Again, I'm interested, as Beverley, I'm interested in things that are inexpensive, accessible, and scalable. And so that's really what we've been focused on. And I think it's something worth considering. So, with your permission, I'll bring it up with the team and see if they've already addressed it and thought about it. And perhaps it's something we might be able to do. I don't know if inhaled heparin is approved here by the FDA. I don't believe it is. So it would sort of fall into that same phase 2 trial approach, but we've done that for other things. So I'll ping someone about it later today.
Thank you. I mean, we are a global community, and if one country cannot do something, then maybe another should try.
Yeah, I might ask
sorry.
I might spread this question a little larger then and say, could we try this in places where there aren't other clinical trials going, places that perhaps have less agenda setting that happens at a top level, and people at a local institution or in a local city or state might be able to facilitate it better. And of course, I have family and I grew up in India, and so I think about that as well. And whether places like that or Malaysia, the Philippines, South Africa, you might be able to, the numbers are large enough there that if there's an intervention that's fairly innocuous and inexpensive to put into place, could you really, in a scrappy way, gather enough data points to be able to, gain support for a larger trial?
Quentin?
Yes, I think that's a very valid point that Yogen makes. I must actually admit that there are efforts all over the world to look at nebulized heparin or heparins, and are in various stages of, I think, application, and some are also underway. So Frank Van Haren and Barry Dixon, who are two intensivists from Australia, and I'm not sure whether you've come across them, but they've made an excellent effort to get a global study going, and that's basically the Charter Meta trial. They've met with some successes in some countries, but again, we need quality data. And I agree with you. I mean, it might not be a full-fledged randomized controlled study, but even if we've got some data that we can publish or access to actually show the clinical benefits of heparins, I think that would be very useful. But saying that, it's not very easy. I mean, there's a few studies going on in South America and Brazil, and I don't know if you're aware of these, but they're looking at nebulized heparin. They're looking at systemic heparins, subcutaneous and intravenously. I think in India it's going to be difficult with what's happening, and the red tape is tremendous. Because I was trying to get a study done, which was essentially an audit, and I've only just got approval for it. So I mean, I agree with you. I think we need to make more efforts to investigate this drug, because I think it will lead to considerable benefits. It has great implications for a future pandemic. Okay. And I can say this also from the lab that I work with. We've done a lot of work in viruses and other pathogens. And because it's a crude molecule that's is not really investigated intensively, I think it provides endless opportunity. So I think that, yeah, I would totally support.
There's a question in the chat box from Helen Philippou, who's a professor of experimental medicine at Leeds. And she's saying, to date, is unfractionated heparin considered to have greater potency than the fractionated heparins?
Yes.
Quentin, Andy, it is.
Yes.
Yes.
So, and I think I probably briefly alluded to that in the chat, and the reason being is unfractionated heparin is a crude molecule, and for those who know about purification of histones, and this is going back decades, unfractionated heparin was used to purify all these very highly charged molecules, because it was so good at mopping up all the other impurities. So unfractionated heparin has its benefits because it's crude and it can bind to a lot of stuff. However, I think when we talk about modified heparins and heparins that are built for purpose, and there are extensive libraries that exist of the same, they do have effects. Now, even the cell invasion paper that we published, and that's the one that Andy put up as a preprint, it showed that unfractionated heparin was superior to low molecular weight heparin. And that's very easily explainable, because unfractionated heparin will stick to anything. So, but that is where I would say caution needs to be exerted, because unfractionated heparin and its use is a double-edged sword. We don't know enough about it, and while it might be useful in this present pandemic, if it is used, I think we need more research into this.
Okay, so there's a cracking question in the chat. Listen to this. This is a different avenue for it. It's from Odi Okariki. Could there be any benefit of inhaled heparin in patients who go on to develop long COVID, theoretically speaking? How are you going to answer that?
Who would like to answer that?
Well, I'm gonna defer to my experts here.
Well,
sure.
I mean, I think the issue is that we don't understand the lung dysfunction that exists in long COVID. So we have quite a lot of people who are very breathless and that they don't have any evidence of pulmonary emboli, and they have some evidence perhaps of dysregulation of the ACE2 receptor in the lung circulation, maybe permanently damaged by COVID. And I know that there's some research going on in London around that. So the answer is possibly, but as we don't really understand the breathlessness in long COVID, I think we have to wait and see.
Beverley, can I say,
yes.
there is sometimes persistent fibrosis, is that right, in the lungs?
Yes.
I mean, and this is something that there's not enough evidence for, but one of the early indications of inhaled heparin was for idiopathic pulmonary fibrosis and cystic fibrosis. And I think there was some early sort of biomarker evidence that it might be useful, but that's unfortunately, I don't think that's really being pursued. So it'd be really interesting to look at from that perspective. Plus, if you have got spike protein or virus still there in the lungs forming weird cells, pneumocytes, whether that can stop that sort of replicative cycle, via the ACE2 binding, etc. Plus, if there's ongoing inflammation, it might be useful. So possibly, but yeah, it'd be a great thing to look at. Yeah.
Okay, so I think we need to wrap up. I'm just going to give Yogen another opportunity to talk about something that isn't heparin. Yogen, do you have any final thoughts? Where are you going with your research? What are we going to think about it in a year at the ISTH in London, which we're expecting you at?
So thank you for that as well. It's very interesting to see the heterogeneity in individual responses to this infection. And it's not just age-based. What we're seeing now in the States is a lot of 30 and 40-year-olds that are in the ICUs. And so there is something, it may not just, it may be viral, the amount of the inoculum, the size of the inoculum. It could be other immune risk factors that maybe are otherwise cryptic and we don't know those, and we're uncovering those here. It's quite possible that many of these, many of the findings we have with heterogeneity in both people getting sick and the response to the illness could be tied back in some ways to dysregulated immunity and in autoimmune responses. And I think we have a very unique opportunity here to be able to really understand immune biology in human disease that may be helpful for us for the future. So for us, Beverley, you asked what we're thinking about next, and it's really this question: trying to understand what drives the B cell compartment to really unleash some of these bad humors, and whether if we can understand that better, we may be able to affect other diseases like lupus and antiphospholipid syndrome as well as some of the more acute illnesses.
Okay, well, I think that's fantastic. Thank you, all three of you, for a really fascinating and fun session. And I wish you all luck with your research, and fingers crossed that there is a grant for Quentin out there for inhaled heparin at some stage. And Yogen, good luck with all your future work. And thanks.
Thank you. It's been a great pleasure.
Okay, so bye everyone.